Well, it's hard to believe, but here we are at the 10th video already in the semester's course on the diversity and evolution of insects. It won't be too long before we go diving headfirst into each of the insect orders, and I like to, one of the goals is to understand the pattern of evolution within those groups and how they have acquired the characteristics that they do. And all of that presumes that we have a phylogeny to work with. And a lot of the phylogenetic work that's being done these days goes under the name of phylogenomics instead of just phylogeny, with the implication that with the genomics in there that they're using whole genome sequences. And so I want to be sure that we all understand, once we get to those, of where those analyses come from, what the data are that underlie those analyses, and how they get there, and so we can better analyze them and critique them in a positive sort of way. So whereas so far I've spent a lot of time talking about concepts and modes of analysis, today's video is mostly going to focus on the data itself, where the data for phylogenomic studies comes from. And of course, it's primarily from molecular sequences. So let's dive in here. I'm going to start out with things that I presume, I hope, I pray, I'm sure is pretty much review for everybody, and I imagine some of you even have some extensive experience with some of the techniques we're going to talk about. But let's start out with the basic dogma of biology, one of the many probably dogmas of biology, and that is the sequence from going from DNA to express proteins in an organism, in a cell. So we all know that DNA, deoxyribonucleic acid, is composed of 4 nitrogenous bases, phosphate groups, et cetera, arranged in a double helical structure, and that in modern organisms, almost all, except viruses and things like that probably, the DNA is the genetic code that is somehow translated into what ends up becoming the final organism. So there are lots of pieces to that, many of which we don't understand fully. Of course, there are the structural elements, the proteins, the RNA that is made from ultimately following the instructions of DNA. And then there are control elements, feedback loops, all sorts of things that go on that we have still a lot to learn in those areas. But the basic dogma, basically since, like, well, I don't know, probably the 1960s or so, is that we start with DNA, and then that DNA is transcribed to produce various kinds of RNA. Ribosomal RNAs that go to make up the ribosome, messenger RNAs, which we're going to focus on here in just a second, transfer RNAs, and then there are all sorts of new RNA species, if you will, that are being discovered these days, and we're trying to figure out what they're doing. But to take the next step in that dogma, we have first the transcription of DNA to RNA, and then the translation of that RNA, specifically the messenger RNA, into protein. And so that's the basic sequence. And this is just the graphic. Where did I pull this from? One of the U.S. government slides to just illustrate this process, going from transcription of DNA, so double-stranded DNA, to pre-RNA, which then gets processed. You know, they have poly-A tails that get pulled out, and eventually, I guess the poly-A tails do get transcribed too, but the messenger RNA can come out. Actually, you know, when you get into something like the mitochondrion, you can have multiple coding segments transcribed into a single RNA, and then somehow they later get chopped up into the relevant proteins that those go into. Anyway, we have the pre-RNA, exons and introns in there. We get rid of the introns. That's one of the major elements in processing the messenger RNA. It then leaves the nucleus, and is translated through the ribosome. This sort of, I guess it's labeled down here, this amorphous hat thing here is where the RNA is translated from nucleic acids into amino acids in the growing protein chain. And so our data can come conceivably from all of these steps. We can have data that's derived from DNA. We can have data that's derived from any of the RNAs, but primarily messenger RNA and ribosomal RNA is what people have been using. And in some cases, even directly the amino acid sequence of the protein, although in most cases that's inferred from the RNA and DNA sequences of what the amino acid sequence of the protein is eventually going to end up being. Get my pointer in the right place. Here we go. So for the past, for the past 40 years or so, the systematics world, in terms of what we have done to get molecular data for phylogenetic analyses, has been dominated by two processes that go hand in hand. And the first, of course, is Sanger sequencing. Sanger sequencing is the method of determining what the sequence of nucleotides are in a DNA molecule. And while there are many sequencing techniques that have been devised, Sanger sequencing is really the one that still occupies a major position in that whole field. This was invented in 1977, only 1977, by Frederick Sanger, who was a British biochemist. He was one of the few people, I think there are a grand total of 4 people who have won multiple Nobel Prizes. Sanger in the early 1950s was responsible for sequencing insulin, and then for the development of Sanger sequencing, which basically generalized sequencing and made it something that almost literally anybody could do, won a second Nobel Prize. And so this sequencing technique is called sequencing by synthesis. And basically what it involves is taking, copying DNA using a DNA polymerase, and providing the nucleotides, the deoxynucleotides that are the building blocks of DNA, but adding to that mix of deoxynucleotides, we add dideoxynucleotides. Now, dideoxynucleotides, because of their chemical structure, are unable to form a bond with a subsequent nucleotide. So whenever a dideoxynucleotide gets incorporated into the growing DNA molecule, then that stops any further elongation of that chain. And so basically what you do is that you link dideoxynucleotides with a fluorescent marker, a different color for each of the 4 dideoxynucleotides that it could be, and so that you can tell what the last nucleotide that was added to that growing DNA chain was. And then through basically a process of electrophoresis, you separate them by the size of the DNA molecule, and then just observe with a laser how the chain fluoresces, and thereby understand what the last nucleotide that was added to that chain was. In the real olden days, of course, they didn't have the fluorescent markers. What they used were radioactive markers, which were problematic from a variety of aspects. And so the sequencing, people were doing sequencing in their lab in the 80s and in the early 90s, and using radioactive markers had to adhere to all the safety protocols of the people concerned about radioactive safety. And so that was a pretty stringent thing. I took those safety classes once. I never got into it myself, but I did take the safety classes, and they're hardcore. They were tough inspectors. Now, if you've Probably many of you have done this or at least seen this, but if you haven't, go in the right direction here. There we go. This is it's called a trace file for a sequencing run, a Sanger sequencing run, just to show you what this looks like. So let's see, if we start up here, basically this loops around, so each of these rows actually should be connected end to end to form a nice long row. But here we have the beginning of the DNA run. So basically it's moving through a column and it's sorted by size. So the shortest DNA chains come out first, and then the longer and longer come out later. And as they pass through a detector, a laser energizes the fluorophore, I think that's what they call it, to which the dideoxynucleotide is attached. It fluoresces, and the wavelength at which it fluoresces tells you which nucleotide it was. And so here we have the beginning of the run, and beginning of the runs and the end of the runs are usually pretty much garbage. I've got the end of the run hidden there behind my little screen, but you can see beginning of the run, there's some confusion here, and this line underneath is an automated base calling, is the results of the automated base calling. That is, the detector measures the wavelength of light that's being emitted from the fluorescent dye and saying, well, that's an adenine or that's a thymine. But you see here at the beginning, we're getting all sorts of different, we start out not getting any or very little fluorescence at all. Then we start to get some, but there's multiple wavelengths showing up here, and in this section here you can see many different wavelengths. So these ends here at the beginning mean that the automated base caller was unable to determine which bases were present in those positions. And so this is one of the reasons why you probably found that when you got sequences from GenBank for your phylogenetics project, that they all didn't start in exactly the same place, because there's no set amount of time before this sequencing run sort of settles into a very regular pattern like we see down here. And so you got to throw this away because this is ambiguous garbage, and you might not be too interested in what's going on in here as it kind of settles in. But once it does, you can see that we're getting calls at almost clock-like regular intervals along here, and the amplitude of the fluorescence increases through time. We get a little confusion in this section here, where we've got, I don't see the key, what is it, for what, that's T, the green line here, so that's thymine. So we're getting thymine showing up along with, what is that, adenine at that position. So we get a little bit confused here. This background image shows you the assessed quality of the read at that point. So here we have low quality reads, but then they jump up here and they stay pretty high through much of the rest of the run. But as we go along, you see you have regular peaks, and it's typically very clear which base is showing up at which point. So let's see, what else did I want to say about this? As we get toward the end, we start to see some background noise showing up, and the quality decreases. I don't know if we actually get any ends at the end here, but it definitely do there behind this screen there. So that's what a trace file looks like. And so this one goes from, this is the start one, and we go to, if that's 600, 610, 620, 630, 640 base pairs, of which we're going to have to trim. If it were up to me, I would probably trim at least the first 30, maybe 32 bases off and say that's not usable. And I would have pretty good confidence here, except for that spot. And then I have great confidence that these are correct. And then it's going to start to deteriorate down here a little bit. And so somewhere I would cut that off. So if that's 620, 630, 20, 30, 40 base pairs total available, then I've got a good 600 or so or at least 550 base pairs that are quality data that can be used for my analysis. So Sanger sequencing is great, but you have to have the DNA to sequence, right? And it takes a fair amount of DNA in order to do the sequences. So how do you get that DNA? When you extract a specimen, I mean, the insects that I studied, they're a millimeter long. And so when you do an extraction, you can get DNA, but you're getting a very small amount. You're not getting anywhere near enough DNA in a whole body extraction of that organism in order to sequence anything there. So we need more DNA. We need more copies of the same DNA we had. And I'm sure you know that leads us to the partner of Sanger sequencing. And this partnership between polymerase chain reaction, or PCR, and Sanger sequencing literally revolutionized not only systematics, but so many other fields, and really elevated molecular biology to the position it occupies today. Most people don't talk about this. We talk about Sanger sequence when you know who Frederick Sanger is, but not many people know about the invention of PCR. The idea was conceived by Kary Mullis, and the process was conceived and invented around 1983. So, at least from my perspective, not that very long ago. Mullis was awarded the Nobel Prize in chemistry in 1993, 10 years later for this. He died just 2 years ago, I think, something like that. But this is what really transformed, enabled us to use Sanger sequencing in an everyday lab experience. You didn't have to have special DNA labs in order to produce the quantity of DNA necessary for Sanger sequencing. And basically, I mean, this should be review for everybody. PCR works by, you have two primers. One at the beginning of the sequence you want to know, and one at the end of the sequence. And all the primer is a short stretch of DNA, 20, maybe 25 base pairs long. There's some variation in how long they can be. And the idea is that you're going to, the primers will bind to the template DNA, and then you will start to replicate the DNA using a DNA polymerase. And at regular intervals, you will heat that chain up, the DNA molecule, which causes the two complementary strands of the DNA to separate, and then you restart the whole process. And so every time you do this, you're basically doubling the amount of DNA of that section of interest, with a little bit of slop in the early stages, but you end up basically duplicating at every step the DNA between the positions that those two primers bind on the template, the target DNA, into enough copies such that you can then use that as input to Sanger sequencing. Of course, when you're heating and separating those DNA strands, you don't want to be degrading the DNA polymerase you're going to need. And so the polymerase that's been used is something called TAC, which the name itself stands for Thermus aquaticus, which is an archaean. So what we used to call Monerin, was originally discovered in the pools of heated water in Yellowstone Park. So the TAC polymerase that's now sold all over the world is actually comes from a natural product that was your, as an American citizen, it was your heritage, your natural heritage, coming from those hot water pools. So as I said, the combination of PCR and Sanger sequencing dominated biology, and in a lot of ways it transformed systematics for well over 40 years. And I don't mean to imply by that the era of Sanger sequencing or direct sequencing is over. that there's still lots of studies that used the combination of PCR and Sanger sequencing, lots of good studies that come up with very valuable results. The other thing that this does, what PCR enabled, was for a dramatic decrease in the price of sequencing. So now the cost varies widely depending on which sequencing company you go to, and this has now been commercialized so much that, frankly, I don't think there are very many labs at all left that actually sequence their own DNA. In EEOB, I know there's a lab that still has a sequencer is a piece of software, a sequencer that some people like to use in order to test whether the primers that they've designed using computer programs will actually work in order to amplify the sequences they want from their own DNA. But by and large, this is all farmed out to third parties. At one point, we were even sending our DNA to South Korea to be sequenced. And so there's lots of labs all over the country that are doing it, and their prices will vary, and there's sales and specials and all sorts of things going on. But just roughly, we're talking somewhere on the range of $2 to do a sequencing run. Nobody, of course, does a single sequencing run. It's typically, 96 well plates, or you're doing 96 samples at a time. So, $1,500 to $2,000 for a 96 well plate. But each of those, if the reaction works correctly, can garner you somewhere, in a typical primer pair, somewhere between 500 and 600 base pairs. And you do the math, and that's something like 0.3 to 0.4 of a cent per base pair. So, if you think about that, then that's data that at the beginning of my career, that was really hard won and very expensive data to gather. And now it's well within the price range of most labs. But the world is changing. And of course, you're familiar with next-gen sequencing. I'm not going to get into the underlying technologies in next-gen sequencing. But I do want to give you a sense of the scale of the change that's going on. This is the so-called next-gen sequencing. And basically, I think we're now in third generation or more in terms of sequencing technologies along this line, has resulted in a just mind-blowing expansion in the sequencing capacity. So instead of being able to get, if you're lucky, 500 to 600 base pairs in a sequencing run, now you can literally get billions of base pairs in a sequencing run. There are a couple of different strategies for this, and this is that second versus third generation sort of approach. The older one, what I've kind of tongue-in-cheek here called destroy and rebuild. Illumina is the most common technology, a set of technologies, I should say, that people are familiar with. Roche 454 sequencing was one that I used years ago. It's kind of fallen out of favor these days, and I don't even know if the company is still active anymore. But the basic idea behind both of those is that you take your starting DNA, and basically the first thing you do is you break it all into little pieces through shearing. You can either do that enzymatically, or more commonly, people would subject a DNA sample to a sonicator, so you're hitting it with sound waves to shear the DNA into small chunks, and you want them a particular size for everything that you're going to do next. So once you have all those small chunks, you do some internal amplification, and then select the size you want to sequence, the size chunks, because they come out in all sorts of sizes, but select one size range, and then sequence those. So you're sequencing many small pieces of DNA. How small are these pieces? Well, again, technologies vary. When Illumina started out, you would be getting a 50 base pair read. So instead of 500 base pairs that you could confidently get from a Sanger sequencing run, now you're getting 50 base pairs. That seems like a retrograde step until you consider that you're not just getting one run as you would, one sequence as you would in a Sanger. And so instead of getting one 500 base pair sequence, you're getting millions of 50 base pair sequences. And so then you take those sequences and find where they overlap, where the DNA sequences match and they overlap, and you start to piece all of that back together. So this is shotgun sequencing. Blast it all apart and then try to reconstruct it after you know what all the pieces are. The newer technologies, they're a little bit old even at this stage, and this is a rapidly changing field, so I'm certain that I'm not up on all the latest and greatest things that are coming out of these companies. But an Illumina MySeq platform, which is one we commonly use, for a single run will give us somewhere between 24 and 30 million reads, and each read is a 300 base pair paired end read. That is, I've got 300 bases, and then I've got 300 bases over here. I don't know what's in the middle, but I know that they're connected to one another. So I've got 24 to 30 million of those, which then I can then piece together. And a single MySeq run, again, the prices vary, but roughly between the cost of preparing the library and actually doing the sequencing, you're talking $1,500, somewhere in that area. And so doing the math on that, I don't even know what those numbers are, but I know the first one, that's 7 zeros before the 7 fives. So you're talking about tiny fractions of a cent per base pair for a MySeq run. A HiSeq run gets you more reads. They're shorter, but you get more of them. So instead of 24 to 30 million reads, you're getting 3 billion. So what is that? That's 100 times, 100? Yeah, 100 times more reads. They're a third shorter, so 100 base pairs instead of 300 base pairs. So you're getting much more data there, even though the reads are shorter. And whether you want to do the 100 or the 300 base pair kind of depends on what you're using, what you're doing with the sequencing. If you want to, for us, we want to reconstruct whole genomes, and so there I like the longer base pairs so that I have more overlap potential overlap between the pieces. There are now also some long read technologies. PacBio and Oxford Nanopore are two of the prominent names. In comparison to something like Illumina, these technologies have a higher error rate. And so while you think that might be a damning negative, the positive side of these technologies is that instead of getting a read that's 100 or 300 paired end read, you're getting potentially reads that are 100,000 base pairs in length. And so you might have a higher rate of error in there, but you're getting much longer reads, which enables you to circumvent the issue of reconstructing those small pieces and finding the overlaps between them and correctly finding the overlaps between them. What most people recommend is a hybrid approach, is that you take advantage of the Illumina's ability to have a higher accuracy in their reads with the longer reads of something like Oxford Nanopore, and then you combine those two things and you use the Oxford to scaffold your Illumina reads on there and get the best of both worlds in that process. So these are techniques that are not, I mean, they're not cheap, but they're well within the financial capacity of many labs to do. And so it's possible, and we've done it several times, to sequence the entire genome of the organisms that you have of interest. Of course, you have to have specimens of quality sufficient quality in order to be able to do this. And for these long read technologies, you really need high quality DNA. So stuff that comes from live cultures is best, and stuff that you can get in quantity. So being able to pool multiple specimens together in order to get the total quantity up to where it's analyzable in these situations. Okay. So given the capacity to sequence entire genomes, well, why the heck don't we just do that? Why don't we just sequence entire genomes and compare them and construct phylogenies? Well, there's a lot of issues that go into that. One concerns genome sizes, and the size of genomes, interestingly, varies tremendously in different groups of organisms. Just within insects, the smallest genome, at least the last time I looked this up, the smallest genome is that of Belgica antarctica, which you Denlinger Lab veterans will know is the flightless midge from Antarctica. Its genome is only 99 megabase pairs, so 99 million base pairs. Sounds like a lot. It's actually quite small. Belgica has almost all the genes that other flies do, but it's gotten rid of intergenic regions. Its introns are shorter, so the whole genome is compacted. Why the heck that would be, we can only speculate at this point. In contrast, if you're interested in studying grasshoppers and locusts, you've got a real problem, because their genomes are monstrously larger. The genome of Locustra migratoria, the migratory locust, is 6.5 gigabase pairs. So 6.5 gigabillion base pairs, as opposed to 100 million base pairs in Belgica. And it's not only the magnitude of the size of the genome that's the issue with something like Locusta, but it seems to have acquired these monstrously sized genomes through a process of genome duplication. So if you're trying to sequence the genome of those things by taking the entire thing, blasting it into tiny pieces, and then reassembling it, well, you don't just have one copy of everything, you've got multiple copies of everything. And so how do you know that you've pieced this together correctly? Because there's going to be a lot of similarity between those copies. So this is a real technological issue that has made it hard for people studying Orthoptera to do some of the other techniques. In my own lab, the genomes we work with are on the order of I don't know, 150 up to 300, 350 megabase pairs. So quite tractable in terms of the being able to do the sequencing and get enough copies of the sequence, what they call sequencing depth, to be able to reconstruct it from the small reads, and to do so in a manner that is computationally effective. In other words, we don't have to sit around for 6 months waiting for a computer program to complete the process of trying to reconstruct these things. It's a fascinating area of genome assembly from these short reads. There's a lot of art, there's a lot of science to it, there's a lot of math, but there's a lot of art into doing these things correctly and well. But hey, even having said that, after you've got a whole genome for a set of organisms, how do you compare them? We know that organisms differ in chromosome, in their chromosomes. So the number of chromosomes, the size of the chromosomes. So already the genome is broken into discrete packages that might be difficult, perhaps impossible to match. We have chromosomal inversions. Take a piece and flip it over the other way. Duplications of parts, repeating elements. We have intergenic areas of variable sizes. whether that's just nonsense DNA or there's something going on there in terms of RNA, or who knows, there may be yet other things discovered that's going on in these intergenic regions. Those are going to vary between organisms. The number, size, and position of introns within protein coding genes is going to vary. In other words, it ends up being a very difficult exercise. I think I can safely say next to impossible exercise to simply take a whole genome sequence, align it, and have a valid comparison amongst species. They would have to be very closely related and have basically the same genome metastructure in order for that to happen. And that's just not what we find in most organisms. So we're really looking for orthologs, right? We're looking for, well, it doesn't have to be protein coding orthologs, but stretches of DNA that we can validly compare across organisms. Maybe some are closely related species. That'll be relatively easy. But if we're looking at across all insecta or across all arthropods, it's going to be much more difficult to find these areas that we can validly compare. So what I'm going to spend the rest of this video on is talking about some of the techniques that people have devised and you're going to be reading about in the rest of this course to be able to take this potential treasure trove of information in the genomes of these organisms and actually do the comparisons. So the first one I want to talk about, well, they all fall under the rubric of what I'm calling reduced genome technique. So instead of looking at the entire genome, we're going to concentrate our attention on a reduced set of targets within that entire genome. So the first technique I want to describe is something called ATRAM. And I'm prejudiced. I'm going to talk about this one first because this is one of the ones that we've chosen to use here in my own lab. So ATRAM is a protocol developed at the University of Illinois, and it stands for Automated Target Restricted Assembly. So you can figure out what the automated part means. The target restricted assembly means I'm going to restrict the parts of the genome that I'm going to put together from all of my millions of short reads to some target that I have identified in advance. So the first step you need to do is to identify those target sequences, to work, to find, to hypothesize and hope they exist, orthologous genes amongst the set of organisms that you're studying and that you want to analyze their interrelationships. There are a number of tools available to do this, to find orthologs. So typically you would start out with maybe one or a few whole genome sequences that you've assembled and that you've annotated and found what genes, and then look for orthologs in other organisms. And then you want to make sure that these orthologs occur only in single copies, because if you have multiple copies, that is paralogs, then you might end up comparing paralogs in their sequencing in your alignment and get erroneous results as an outcome. So identify target sequence, single copy. What we targeted were single copy nuclear protein coding genes. And then you take your excuse me, your MiSeq or HiSeq reads, so all those millions and millions of paired end reads, and map them to the sequences. In other words, you would take a read and compare it to all the sequences in your target set and find which one it met, which one, if any of them, your read matches. If it doesn't match any of them, throw it away. If it does match it, then you kind of assign it to that target sequence. And in doing so, you do this through iteratively. You do a little, then you go through again a second time, and so now you're having a read that might match with a read and a target sequence. So basically you're building up the gene as much as possible. Once you're done, you remove the introns. There are programs that do this, and then go back to the same kind of stuff you're doing in Mega. Align and analyze, except you're doing it on a scale 100 or 1,000 times larger than you're able to do by hand in something like Mega. Here's a flow chart showing the process. So basically up here we have the input, which is a bunch of millions and millions of paired end reads that we're going to compare to our target sequences and see how they line up. And so over here, this is an explosion of that. Here we are going through multiple iterations. This is the first iteration. Then you do that again, run them all through, find out how they line up, and gradually build up the target sequence until, you know, if you're lucky, you get the whole target sequence here in the final assembly. You're not going to get them all. Some are not going to be present. There's some ambiguity. You're going to have gaps and holes, but that's okay. Hopefully, the underlying assumption is that the volume will compensate for the shortcomings in the reconstruction of the individual genes here. And then once you have those thousands of genes, you do your analysis just as you're familiar with. Okay, second technique. So that's the ATRAN protocol. Second technique is transcriptomics, which I'm sure many of you are familiar with. In transcriptomics, basically, you are, instead of sequencing the DNA encoded in the nucleus of an organism, or the mitochondrion, or the chloroplast, or the apicoplast, or whatever you have in the organism of interest. Rather than doing that, what you're doing is sequencing the messenger RNA that cell, that organism is transcribing in its cells. So the first step is to take your organisms and extract the messenger RNA. Then use reverse transcriptase to convert that messenger RNA back into DNA, now called cDNA. And then using the same protocols for Illumina protocols or whatever, sequence that DNA. So you're not sequencing the RNA directly. You're transcribing it back into DNA. DNA is much more stable and easier to handle and will last. And plus the sequencing techniques. I guess there are some that you can sequence RNA directly, but it's more difficult to handle and more liable to break down and you lose your information. So you transcribe it back into DNA. And then if you're using MySeq or HiSeq or some Illumina protocol, then you've got the millions of reads. You're back in the position of having to assemble those small pieces into the longer DNA from which they originated in the first place. Transcriptome is valuable in that it's giving you a snapshot of what the cells in that organism are transcribing at the moment that you extracted the DNA. So it's telling you something about the physiological condition of the organism. And typically you'll get data for thousands of transcripts. And these are all protein coding genes. They've already all had the the introns removed from them. So you're only dealing with exon sequence. So they're directly comparable. You could translate them into protein if you want to, instead of or in addition to analyzing the phylogenetic signal in the DNA, you could do it for the amino acids in the protein as well. And so this is a technique that is quite widely used. It's relatively straightforward. The difficulties with it are, first of all, that it requires you to extract the RNA. And in order to get enough, especially if you're doing a transcriptome from a small animal, you need lots of these animals. And so it's not something that is well done, I think is the way to say it, on a single organism. Of course, they can do transcriptomes. They do transcriptomes on single cells these days, but that has its drawbacks as well. And one of the issues is that if you're getting a very small amount of RNA, and then you have to basically you reverse transcribe that and then amplify that, you may be losing genes along the way simply because they existed in your initial sample in very, very small numbers. Or perhaps they didn't exist at all because the organism at that point in time in its life was not transcribing those genes. So, some things are transcribed only in the larval stages. Some things are transcribed only in the pupal stages. Some things are transcribed only under certain environmental conditions. And so the experience and the stage of the organism that you are extracting the RNA from ideally should be controlled so that you're really going to be able to compare things directly. Because it's using RNA, this is not something that you can really do effectively on preserved organisms. So if you go out and you're collecting specimens in traps or something like that, as many entomologists are going to do, then the RNA is not going to be worth sequencing once you get back to the lab. The DNA is still probably good, especially if you take good care of it, keep the stuff in high concentration ethanol, or if you can, keep it at low temperatures, things like that. So transcriptome is not going to be particularly valuable for field collected specimens, which then, of course, limits its applicability to things that can be cultured in the lab, which most species cannot be. Another difficulty that you have to keep in mind is that some genes, some protein coding genes, of course, they're all composed of exons, but some of these genes can mix and match the exons in different ways to produce different variants, what are called splicing variants, different variant proteins from the same DNA sequence. So it might use exons 1, 2, 3, 5, and 7 to produce one protein, and then exons from the same DNA sequence might use exons 1, 2, 3, 4, 6, and 7 to produce a different protein. In your transcriptome, these are going to look different, and the only way you'll be able to tell if they are just splicing variants or actually different genes is to have access to the genome, to map those back to the genome and see where they show up. Do they overlap in the genome, or are they totally separate places? So there's some issues. There's some issues with everything, right? Okay, let's see. So that's transcriptomes. The next technique is next two techniques are very similar in many respects, but they go by different names, and we'll see why here in a second. But they started out being referred to as anchored hybrid enrichment. So basically the process here is that you had a set of organisms from which you want to get the sequences for a number of regions of the DNA. So you've got a number of target sequences that you're after. How many? We're usually talking 1,000, 1,500, maybe even more different sequences, different target regions of the DNA from which you want to get the sequences. And so the strategy here is that well, you need a model organism or a transcriptome or something like that, the genome of a model organism or its transcriptome, to know what those sequences are in at least one case, and then design probes. And all a probe is, I've seen them both DNA and RNA. It's a relatively short DNA short nucleic acid sequence that matches the target region in the organisms you're going to get data from. Either matches entirely, or they need to match pretty closely. The probes are, short, 120 base pairs, something like this. And so you have a long sequence, and you have short sequences in the probes that match parts of it. And you design these probes such that they overlap, and so that you can, in theory, get that entire stretch of target DNA, probes binding to it throughout its length. Now, the probes, the DNA or RNA sequences, are biotinylated. That is, they have the molecule biotin attached to them. Biotinylated probes can be bound to magnetic beads. So the idea is that you put your probes in to a mishmash of DNA from the organism you're studying. The probes bind with the DNA that you've already sheared the DNA in small pieces. The probes bind with those pieces through the matching nucleotide sequences. And then you apply a magnet to it and attract all the beads, these tiny, tiny little beads, then go to the magnet. You remove that, throw away everything else, and then release get the DNA that you've bound from the beads. Get that released. Basically, digest the probe away to release, so that it's the DNA you want to sequence is no longer bound to the biotin. Thereby, it's released from the magnet and into solution. I've got an illustration of this in just a second, but let's throw the caveat here in the first place, or early on, that you have to have your probes bind with them, right? And so you've got to know what the sequence is in your target regions, which means you need to have transcriptome, or you need to have a genome done in the first place. So you got to do some preliminary work. And then you have to target areas that the sequences are similar enough between the original organism upon which you have designed the probes and all of the target organisms. And by definition, you don't want the target organisms to have exactly the same sequence as the one you started out with. Otherwise, you've got no information about how these things are interrelated if all the data are exactly the same. So you need some variation, but you don't want too much going on there. So here's a graphic. I thought a graphic might help to explain this idea. So first of all, here's our genomic sample that we shear the DNA, either enzymatically or sonicate them, and then we have designed on the basis of a pre-existing transcriptome or genome, a set of probes, so short, in this case, RNA library. They're calling them baits instead of probes, the jargon difference between the two. And then you combine these two together, and the baits or the probes will hybridize with your target DNA. And then you add the magnetic beads, which the biotin then binds to, that is on the baits. And so some of the DNA baits that have hybridized with your target DNA now are attracted to the magnetic beads. And the rest of the DNA that didn't hybridize can be washed away and removed. You take the DNA bound to the probes that are bound to the beads, and then digest away the RNA so that the probe is gone, leaving you the DNA. Sieve out the beads as you're going along. And then you go through the standard procedures of amplifying those and then doing sequencing, typically Illumina sequencing, in order to, and then reassembly of those in order to reconstruct your original target sequences. And then you compare them to your standard phylogenetic techniques. So that's the general protocol. Again, these probes, these baits, have to be sufficiently similar to your target DNA in order for that binding to take place. If it's not similar enough, these are not going to bind, and that's all the DNA you wanted to sequence is now in this fraction, and it's going to be lost. So that similarity between your probe or your bait and your original DNA, the DNA that you want to sequence from all your the OTUs in question in your study, leads to the second technique, and this is what's called ultra conserved elements. And in many ways, in my mind at least, anchored hybrid enrichment and ultra conserved elements are pretty much the same thing. Part of the reason for the discrepancy is that there was a group at Florida State University that has basically made a business out of what they originally called anchored hybrid enrichment, and I believe their company is called something like Lecture10Phylogenomics. So while ultra conserved elements in principle are pretty much the same thing, this is done by other groups, and so they seem to have shied away from using the same terminology there. Who knows? There may be legal trademark restrictions or something. I'm not sure about the details on that end. But so ultra conserved elements, or commonly they're called UCEs, just the acronym for that, are areas of DNA that across a wide variety of organisms are exactly or nearly exactly the same sequence for whatever reason, and often we don't understand what those reasons are. In some cases you can figure it out, structural constraints imposed by the protein that this gene might be translated into may force it to have the similar sequences, but we don't always know. But they turn out to be present, and people have designed UCE probe sets for wide-ranging groups of organisms. There's a UCE probe set that works for all vertebrates. There's a UCE probe set that works for all Hymenoptera, for example, or at least it works for a wide range of Hymenoptera. Should never say all. So the ultra conserved means that the sequences are the same across a wide variety of organisms. Therefore, your probes will bind with the DNA from a wide variety of organisms. But if they're ultra conserved, that means that the DNA you might sequence from where the binding of that probe takes place is basically going to be useless to you, because there's no variation to tell you which species are more closely related to one another than they are to another species. So the real data of interest is not the sequence of the ultra conserved regions, but of the regions on either side of that. So we have an ultra conserved area of DNA, but it's flanked by DNA on the 3' side and the 5' side, that you're getting more variability, and that's where the data of interest lies. Here's a graphic to show you that. So what do we have here? So imagine we have a chunk of DNA. So this is the proportion, excuse me, of the sequence length. So we're starting here in the middle of the ultra conserved area, and then 50%, 100% in one direction, 50%, 100% in another direction. Here's the UCE. On the vertical axis, frequency of PIS is the frequency of phylogenetic informative sites. Just another way of saying variable sites. We need variation amongst our organisms in order to get data useful for reconstructing phylogeny. So this is the frequency with which that variation occurs in the DNA as we move away. I don't know which way they've got this oriented. Let's say the 3' direction and the 5' direction on the DNA strand. And you can see as you get away from this ultra conserved region where there's a very low frequency, 1%, 2%, less than 5%, there's the 5% line right there, of sequence variation here. Up here, 20% variation on these tails as we get away from this core of the ultra conserved element. So the probe is going to bind with this part, and then as we sequence it and probes bind to probes, we're going to start picking up more DNA on the edges, and that's where our data of interest is going to lie. And so this is a very common technique used to extract from the entire genome areas that we believe will be comparable amongst organisms that have different chromosome structures, different DNA sequences, ultimately, to extract common regions that we can legitimately compare in order to infer phylogenetic relationship. So, what are the issues with ultra-conserved elements? The one that concerns me, and this will lead into the next slide, is that the ultra-conserved area that you have found, which is typically derived from comparing genome sequences, not transcriptomes, this ultra-conserved element may occur within a protein coding gene, in which case it might be picked up in a transcriptome, but it may equally as well occur in intergenic regions, so areas between, perhaps even introns, although typically they're quite variable. But many of the UCEs occur outside of protein coding areas, and so then you get into the question is, when I'm analyzing these data, I don't know what I'm sequencing here. I suppose in some cases I could go back to a genome, map it out, and see where it falls. But sometimes it'll be an intergenic region, sometimes it'll be a protein coding region. It's going to vary. And if it's varying between a protein coding region and an intergenic region that is not transcribed and ultimately translated, then should the same DNA model of evolution that's going into my maximum likelihood analysis, or my Bayesian analysis, should that same model apply to both of those areas? Or am I getting a model mismatch and therefore the possibility, the likelihood of getting misleading results? So with that, I want to introduce another couple of ideas, not to go into them in too much detail, but let's just go through the analytical workflow for the data that comes out of, well, it could come out of any of these techniques, from ATRAM, from transcriptomics, from anchored hybridization, from ultra-conserved element data. So the first thing you need to do is to align your data across your organisms. So if you have your target data sets, your target DNA sequences, which you have defined in advance in ATRAM, UCEs, or anchored hybridization, then you separate those out and align them using the standard multiple sequence alignment techniques that you've already played with. In the case of a transcriptome, then you are separating them, and they're going to be you're going to be able to recognize a lot of similarity there in their sequences just by sorting them that way. And of course, if you have access to a genome, you can also reassure yourself that you're comparing orthologous sequences. Anyway, you align them. Then you analyze your alignments using the Akaki information criterion or the Bayes information criterion in order to determine what the best model of evolution there that balances the number of parameters that you have to estimate from your data with the what am I forgetting here? That gets you the best model of evolution that works for the data that you have. So simplicity or generalizability versus the number of parameters that you have to estimate. Now, it's quite possible that not all of the DNA is evolving the same way. And so there is a technique that one can use that's called partitioning your data. Basically taking your data and breaking it up into different chunks and analyzing the different chunks under different DNA models of evolution. So instead of having all of your data being a GTR plus gamma DNA model. Perhaps some of it, even something as simple as Jukes Cantor would suffice. And so you divide these up into what are called partitions. You group them together. For example, what kind of partitions? Why would DNA evolve differently in different organisms and different parts of the genome? Well, perhaps some DNA that you have extracted and sequenced corresponds to ribosomal DNA, and you might want to analyze that separately from protein coding DNA. Another common strategy is to, within your protein coding data, to separate out into different partitions the codon positions within the exons of that protein coding DNA. So you know that in the process of translation from messenger RNA to protein, that the genetic code takes 3 nucleotides at a time. The 3 nucleotides are collectively called a codon, and each codon translates into a particular amino acid. The third nucleotide position within a codon can vary more than the others. For example, if the first 2 nucleic acids, first 2 nucleotides in your codon are both guanine, GG, then it doesn't matter what the third nucleotide is. That codon is going to code for the amino acid glycine. And so there are a lot of what are called synonymous codons, where they are perhaps identical in the first 2 codon positions, but can vary in the third and still result in the same amino acid. That's why synonymous. So that third codon position is freed from constraints of natural selection to vary more than the first 2. To compare, if you change anything in the second codon position, you change the nucleotide in the second codon position, you will always end up with a different amino acid. The first codon position is somewhere in between. It's pretty conserved, but there are some synonymous mutations that can occur there as well. So you may want, because each of these are under different selective pressures, that you may want to analyze your each codon position separately. And in Mega, you've probably seen in the menu, there's an option for you to do that, to analyze these things separately. So you can partition your data if needed. It adds to the computational complexity, but it's certainly done. And then these data, your aligned data that may be partitioned, you've got models selected for the partitions, goes into your standard phylogenetic analysis, either maximum likelihood or Bayesian or both, or into one of the coalescent techniques that I've only briefly mentioned. So at the end of the day, why are we going through all this, for crying out loud? I just wanted to give you an illustration here. So I've hijacked a part of an image from one of Dwayne McKenna's articles on beetle phylogeny to show you the power that doing things in a phylogenomic context can give you. So here I've just chopped out a little piece of the tree, and let's just look at the scarabioids here. So these are the scarab beetles and their allies. And what Dwayne and his co-authors have done in this analysis is to encode on your topology here, and you see that branch lengths are meaningless here, they're all the same, right? To encode with colors their confidence measures in the clades that they have uncovered. And so this, what is that? Is that a magenta, kind of a purple dot there? It means that in the Bayesian analysis, that particular clade was retrieved with a posterior probability of 100%. And in the maximum likelihood analysis, they came up with a bootstrap support value of 90% or more. And what I want to stress here is just look at how many purple dots there are going along here. The vast majority of them are purple dots, meaning that with the quantity of data and the way they analyzed it, they feel that they have basically solved the problem of how these organisms are interrelated. They're getting maximal support on the basis of their analysis for these data. As a matter of fact, Dwayne gave a seminar at Ohio State 5 years ago or so, in which he claimed that now they know the pattern of relationships. For me, as a scientist, I cringe and recoil if anybody ever tells me that they know something for sure. So I'm just willing to say that with this set of taxa and these data and these analytical techniques, these results are maximally, very strongly, highly supported. I'm not willing to go that last step, though. So let's just briefly summarize this. Although the combination of PCR and Sanger sequencing, what's sometimes called direct sequencing, is no longer as dominant as it used to be, it's still out there. It's still quite useful, and there are still some very important papers that are being published using data from them. But now it's possible to get data not from a handful of genes, 5, 10 genes. It's not possible to get data for thousands of markers. I'm using markers so that I can be equivocal about whether they're actually protein coding genes or some other part of the DNA. So markers, DNA markers, thousands of them. Hopefully, many of them are going to be independent estimates, provide independent data to help you to infer the phylogeny of the group that you're studying. While you can sequence and assemble whole genomes, direct comparison of whole genomes across organisms, particularly when they're not particularly close related, is difficult. And so what's typically done is to have one of these reduced genome techniques in order to focus your attention on particular parts of the genome for analysis. In ATRAM, basically you sequence everything and then extract from those sequences the ones that match the markers that you've identified as being interesting. I did have, there's transcriptomes. I thought I skipped it. In transcriptomes, you're sequencing whatever is expressed. So it's always important to do as well as you can to make sure that the RNA that's being extracted is coming from organisms with a similar life history, similar condition, so that they're expressing the same genes. Then we have the anchored hybridization and UCEs, which, as I say, are very similar. Basically, you are extracting only your targeted DNA and then sequencing. So ATRAM, sequence, then in silico extract, in the computer extract. Anchored hybridization, UCEs, extract the DNA sequences and then sequence them. And then that's the input that goes into a common analytical pipeline that we've already discussed. Okay, so that's all for today. We'll catch up with you next week with the next series in this video series. Thanks and bye.